Hey guys, in today's lesson, we are going to be looking at the stages of mitosis. So we have already discussed that the cell cycle includes three sections that are interphase. G1, where the cell goes about its normal, regular, everyday life. S, where the cell synthesizes or replicates its DNA in preparation to divide. G2, where it bulks up, it increases the amount of organelles that it has in order to make sure that it's nice and large and big enough to tolerate cell division. And then finally, our cell is going to go ahead and enter mitosis, and it's going to systematically divide its DNA. DNA is so important for a cell. If the DNA gets messed up, the cell has cancer, the organism has cancer, or worse, sometimes it's not even as long as cancer. I mean, if it's during development and you get an extra chromosome, you might see the death of the organism right away. So it's really important that cell division happens systematically and correctly. After mitosis, then the cell divides up all of its cytoplasm and it reorganizes its Golgis and mitochondria, smoothie R, Rephi R, and it separates them into the two daughter cells. The stages of mitosis are consistent. First, it starts in prophase. So in prophase, we see the chromosomes start to condense and the nuclear envelope starts to go away. In metaphase, the spindle fibers form the kinetochore, and they start to push the chromosomes to what we call the metaphase plate. So this right here is the metaphase plate. You can recognize the metaphase plate when you're looking at a cell because you can see that the chromosomes are all kind of lined up. Then they start to pull away. They pull apart from each other to the opposite poles of the cell or the opposite ends of the cell, and you can kind of see them dragging back. And then in telophase, the DNA spreads out. It forms chromatin, again, from the chromosome. So it unpacks like it was. Okay, so we're going to look at these stages one at a time, starting with prophase. So in prophase, our chromatin condenses into chromosomes. Remember, that's the idea that we're going to pack up, right? We're going to pack up all of our DNA and get it organized so that we move it from one place to another really smoothly with no mistakes. The nucleolus disappears. You may remember that the nucleolus is where ribosomes are made. While we're dividing the cell, we don't also try to make ribosomes. That's multitasking. It leads to problems. So we kind of disband making of ribosomes during cell division like this. So the nucleolus disappears, the nuclear membrane breaks apart. That's because we need to have access to the chromosomes. If we're going to be able to move them apart, we need to make sure that we can access them, get in and be able to connect to them. And the microtubules are going to grow and attach to the centromere of the chromosomes and begin to move the chromosomes around. So all of that happens in prophase, but prophase is really preparation. So P, prophase, for preparation. And so we get our DNA ready, we get our nucleus ready, we get our microtubules ready. Everything needs to get ready. After prophase comes metaphase. And so in metaphase, the spindle fibers have attached to the chromosomes and they have lined them up along the metaphase plate. This can look really nice and tidy. This is in a fish embryo, so this is in an animal cell. And animals have very small chromosomes. Plants, however, have really large mangled chromosomes. You can see here, this is also metaphase. It's not nearly as neat. We can tell that this is a plant cell because we can see the cell walls along the outside. But when a plant cell lines up at metaphase, it still doesn't look terribly clear, okay? You can see the difference between these kind of interphase cells where you can see the nucleolus and you can tell that those cells are not dividing versus this cell that is, but metaphase is more easily recognized in an animal cell than it is in a plant cell. So prophase followed by metaphase, where they line up at the metaphase plate or in the middle, M for middle of the metaphase plate. In the anaphase, they move away or they move apart. Two A words that can help you remember that anaphase, they move away from one another. And when you're looking at them, they even look like these little A shapes as they're being dragged away. So when you're recognizing them by vision, you can tell that as they're being pulled apart, that's going to remind you that they are anaphase. So you can see up here, this is in a plant cell. We can recognize again that this is a plant cell because we can see the cell wall, but also because again, we have those kind of like big gnarly chromosomes, right? They're not nice and tidy in any way. When the cell is pulling apart in anaphase and the kinetochore has been formed and the spindle fibers are connected and they're pulling apart, there are other spindle fibers that did not connect to a chromosome. And instead, those spindle fibers come into contact with one another and they push, as they grow, they actually push the poles of the cell apart. It's like if you were standing next to a wall, you're on roller skates, you're standing next to a wall and you push yourself off. And so you slide backwards. That's kind of the same concept that we're using here. These spindle fibers, one came from one side and the other came from the other side, and they meet and they join, and then as they grow, they're pushing the two edges away from one another. So not only are the chromosomes moving away from one another, but the actual edges of the cell are also elongating and pulling away from one another. So prophase, prepare, metaphase, they line up at the middle, and anaphase, they start to move away. And then we have telophase, and in telophase, we turn back, we reverse all of the things that we did in prophase. So anything that happened in prophase unhappens in telophase. So in prophase, we have the chromosomes condense. So in telophase, we have the chromosomes uncondense back into chromatin. In prophase, we have the nuclear envelope break apart. So in telophase, we have the nuclear envelope form. In prophase, we saw the spindle fibers form. In telophase, we see the spindle fibers dissolve. In prophase, we saw the nucleolus go away. In telophase, we see the nucleolus come back. So everything that happened in prophase unhappens or turns back in telophase. At this point, we also see something called a cleavage furrow starting to form. This is for animal cells. In plant cells, they start to build a cell wall down the middle. But in animal cells, you can recognize telophase by noticing this cleavage furrow, this kind of indentation that starts to form here to let us know that those two cells are starting to pull apart. So that's how you can tell the difference between anaphase and telophase. You can see that maybe it would be otherwise hard to tell here, but because we're seeing this cleavage furrow, we would confidently recognize this cell as being in telophase. At the end of mitosis, prophase, metaphase, anaphase, telophase, we do another division, this time of the cytoplasm, and this is called cytokinesis. So cyto like cytoplasm. So we're going to be moving and separating our cytoplasm into the two different cells. This looks different in animals and plants. In animals, we already mentioned that we get this cleavage furrow, and there are some filaments called actin filaments that kind of squeeze together. Imagine like you were making a balloon animal. You take the two sides and you twist them so that you get the kind of like indentation in the middle. And then eventually they'll be so separated that you're able to just pull them apart into the two individual cells. That's for animals. In plants, it's different because they have a cell wall. What actually starts happening is the cell plate just starts growing down the middle until it forms a cell wall from end to end, and then the two cells have become separated. So cytokinesis looks a little different in each of the types of cells, but it does the same thing. It divides all of the organelles and the cytoplasm so that we end up with two uniform, mostly uniform daughter cells. And remember, the daughter cells means that it's an offspring from the original cell. Not all cells are ready to divide, and this will be our topic for tomorrow. So when next we talk, we're going to be looking at this idea that some cells go through G1 and are healthy and are big enough and are ready, and so they go through G2 and mitosis to divide. But other cells hit what we call a G0 or a repeat, where they just stay in this G1 phase. Those cells might be something like this neuron here. So this neuron, everything in green, is this one cell. And so you can imagine that this one cell, if it divided, probably wouldn't be able to make all of these intricate connections again. And so because of that, this cell would not continue to divide. But we will start back here and talk more about that when next we meet. Thanks, guys. Have a great day.